Now let us talk about the mechanism of action of these fluoroquinolones. Now, as like the previous drugs that we studied, the cephalosporins, the penicillins, and the other beta-lactam antibiotics, fluoroquinolones are also bactericidal. That means they are able to kill the bacteria, right? And they primarily act on an enzyme of the bacteria, which is called as topoisomerase. And topoisomerase comes in two flavors. Either it could be topoisomerase 2 or it could be topoisomerase 4. Now, before we go on to the actual mechanism of action of these fluoroquinolones, let us first talk about a little bit about the bacterial DNA. Right. So let me draw the structure of a bacterial DNA. It is double stranded. So let me just draw it over here, and over that let me put some loops signifying that it is a double stranded DNA. Right. So this is your bacterial DNA. Right. And now what I'm going to do is I am going to show you how these enzymes, which are your topoisomerases, they are going to affect the nucleic acid synthesis. So what I'll do is I'll take a red color pen and I will just cleave out this segment of the bacterial DNA. All right. And now what I'm going to do is I'm going to put out this bacterial DNA on display. Right. So let me draw it over here. And all right. So this is a piece of bacterial DNA that I have taken out from the actual DNA molecule. Now, for protein synthesis to occur, we have to separate these strands. All right. We have to separate these strands. All right. And we have to pull them apart so that the codons that are present within it, like over here, the codon base pairs that are present over here, can undergo the process of protein synthesis. They can undergo the process of replication, or they can undergo the process of transcription process. Now, what happens is, as it is undergoing the process of transcription or replication, what happens is, basically, there is one mechanical error that is going to occur when these strands are going to be pulled apart. Now look over here. Imagine that this is the piece of DNA that I'm going to replicate or translate. All right. So what I'm going to do is I am going to pull these strands apart to make space, right? And when I do that, what will happen to the adjacent strands or the adjacent coils that I have? They will become tightly coiled. Now, uh, as we keep on opening the area of the bacterial DNA that we want to replicate or transcribe, the extreme ends of the bacterial DNA begins to twine very tightly and very closely. And that is actually what we refer to as positive supercoiling. All right. And it is actually a mechanical error that is occurring inside the bacteria. All right. So let me write it over here. It's a mechanical error. And to solve this error, the bacteria has been given the gift of topoisomerases, particularly your topoisomerases 2. And topoisomerases 2, they are also called as DNA gyrase or DNA gyrase, whatever you want to call it. Now, this topoisomerases 2 will solve the problem of supercoiling or positive supercoiling. Now, how does it do that? Now, imagine that you have got this bacterial DNA like this, and it is over here. So this topoisomerase, what it does is it will go and stand over here, right? And this is the topoisomerase 2 that you have, the enzyme. Now this topoisomerase actually has got two functional domains. It has got two functional domains. The first functional domain that this enzyme possesses is called as nuclease. And the second functional domain that this topoisomerase possesses is called as ligase. Now, what do these domains do? Now, as the name itself indicates, nucleases, these are actually your cutter mechanisms. That means one domain or one side of the enzyme will possess, it possesses a cutting scissor type of a thing, which is called as nucleases, right? And on the other hand, this topoisomerase will have a sticky type of functional domain. Now, what will these things do? Now, these things, now whenever the bacteria, whenever the DNA goes into the concept of supercoiling, positive supercoiling, this topoisomerases will go to that actual site and they will cut this part of the DNA. When they cut this part of the DNA, it will cause negative supercoiling. That means it will open up the twinning part or the twining part that is occurring, so that the number of rounds that this bacteria is taken, it will become loosened and it will become less tightened. And when the number of loops is reduced, then the bacteria will require the help of the ligase functional domain of topoisomerase, and it will stick with it. Now, this is actually the sticky part of the topoisomerase, and nuclease is actually the cutting part of the topoisomerase. Now, as and when the bacteria feels that the bacterial DNA is undergoing positive supercoiling, it will require the help of this topoisomerase. It will cut the supercoiling, and when the coils are loosened or are less tightened, it will then stick it up with the help of ligase. All right. So the first thing that topoisomerases do is that they cause a nick in the bacterial DNA, which is called as nicking. And then what they do is they will cause passing through of the bacterial DNA, or the loops gets reduced. And they make sure that the bacteria make sure that they are not changing the gene sequence. They are just altering the DNA loops. So if it changes the gene sequence, that means nonsense codons will be coded and the bacterial protein synthesis will come to a halt. So they make sure that the gene sequence is not altered. And third and the last, what they do is they cause ligation of the cut DNA. Right. So basically what the topoisomerases do is they change the topology of the DNA, and hence the name topoisomerase. Now, since we have understood how the process of nucleic acid synthesis occurs in the bacteria and the use of topoisomerases by the bacteria, let us talk about how the fluoroquinolones will disrupt this. Right. Now, the fluoroquinolones, basically what they do is, fluoroquinolones, they enter the bacteria by passive diffusion. Right. And what they basically do is they actually look like this. They look like a V. All right. And this V, what it does is it goes and binds to the ligase's functional domain of the topoisomerases. So when that happens, only the nucleases part of the topoisomerases will be left free. That means this topoisomerase, irrespective of whether the ligase binding site is free or not, it will go on doing its duty. That is cutting of the DNA at various positive supercoiling places. But it won't see whether the ligase part is free or not. It will go on cutting, and at a time, what happens is this DNA will be fragmented into very short bits. Now let me show you how this fluoroquinolones will go and bind to the ligases. Now it will go and bind to the ligases like this, right? When fluoroquinolones go and bind to the ligases that like this, the, the ligases won't be able to stick up the cut ends of the bacterial DNA. And when that not happens, the bacterial DNA will be fragmented, and fragmented bacterial DNA does not have the property for replication or to ensure protein synthesis in the bacteria. And when there is no protein synthesis in the bacteria doesn't have any option other than to die. And hence, fluoroquinolones are referred to as bactericidal drugs.